Neet biology quiz. Top question from molecular basis of inheritance. Right. Our first question is, at the end of translation, release factors bind to A site, P site, mRNA stop codons, tRNA anticodons. At the end of translation, these release factors bind to stop codons. There are three stop codons: UA, UA, A, G, and another one is UG, A. These are the three stop codons. So these release factors bind to these three stop codons, which terminalize the process of translation and the release of polypeptide chain from the P site. Therefore, these release factors bind to the mRNA stop codons. Next, there are 66 amino acids linked with peptide bonds. Okay, 27th codon is UAU. Now it is replaced as UAA. Now, how many amino acids will be formed by translation? Options are 27, 65, 26, and 63. It's a problem based question. So 66 amino acid means 66 codons. Out of the 66 codon, 26 codon is initially UAU. Now it has been replaced as UAA. So 27th codon is UAU initially. Now it is replaced as UAA. So 1 up to 26. Now 27th codon is UAA. Think of UAA is a special codon, nothing but stop codon. Nothing but stop codon. So during translation, the process will come up to 26, and at the 27th codon, as in the previous question, the release factors come and bind with this 27th codon, and the translation process stops here itself. So you will be getting only 26 amino acids in this new translation. Since 27th codon has been modified to stop codon, therefore only 26. This is only right answer. Lack operon is negative regulation inducible, positive regulation inducible, positive regulation repressible, negative regulation repressible. Right. Lack operon. Lack means lactose. Lac means lactose. Operon means functional unit. Right. See, this is under the control of a regulator gene. Regulator gene called as I. This regulator gene I produces a protein called as repressor. Repressor. So this repressor goes and binds with the operator gene. Goes and bind with the operator gene and prevents the process of transcription. So it is doing negative job. So it is negative regulation. Negative regulation. So on its way, repressor on its way to the operator gene and goes and bind with this operator gene like this. So but when lactose in absence of glucose come and bind with this repressor protein. It undergoes conformational change and frees this operator gene. Therefore, lactose is inducing the process of transcription to digest itself by three enzymes. Therefore, it is inducible. So, lac operon is therefore negative regulation inducible. This is correct. But tryptophan operon is repressible. Tryptophan operon is repressible. This might help you to solve other type of questions. Right. Next, deoxyribonucleotides are added to which position of OH of RNA primer by DNA polymerase? 5 prime, 3 prime, 1 prime. It depends on the respective situation. Right. So it is like this. So this is 3 prime, this is 5 prime, this is 3 prime, this is 5 prime. So DNA synthesis continuously in this strand, DNA synthesis not discontinuously in this strand, right? We will have a look in this strand, right? So here RNA primer is placed. This is called as RNA primer. This is called as RNA primer. RNA primer. Now, so this DNA polymerase is going to so this, what would be this then? 5 prime, and this is 3 prime. This is 3 prime. Now DNA polymerase enzyme, this is a DNA polymerase enzyme, is going to come here and is going to add deoxyribonucleotides to the 3 prime OH of this RNA primer. To the 3 prime OH of the RNA primer, because it cannot, it is RNA primer is like this. The DNA strand should synthesize in this manner, as in the direction of unwinding helicase by helicase. That is why it is synthesized continuously. This is also replication fork is also forming in this direction. The strand is also synthesized in this direction. That's why it is continuous. So it has to add the nucleotides to this 3 prime OH, this DNA polymerase. Therefore 3 prime would be the right answer. So 3 prime is the right answer. It does not depend on the respective position, right? More selected questions are yet to come. To stay tuned, subscribe, like, and share. It's a free entertainment channel. Thank you.